right.
Oh, no, you did great.
That was great,
Run.
right?
That was-
Good
yeah,
morning,
awesome.
everyone. Welcome. My name is Lydia Lazar, and I'm the Executive Director for AcresUSA. And I'm very pleased to present today's presentation and question and answer session on how soil microbes improve the efficiency of fertilizers. It's sponsored by Holganix, a company dedicated to helping growers build soil health, reduce fertilizer use, and boost crop yields through the use of microbial products. Our presenter today is Dr. David Stark. Dr. Stark is a molecular biologist and a biochemist who became the president of Holganix Agriculture in 2015 to lead the company's efforts in commercial development and in research and development. Dr. Stark has 26 peer-reviewed scientific articles and holds 9 patents. The format of today's webinar will be Dr. Stark's presentation, about 30 minutes, followed by about 15 minutes for Q&A. Please type your questions into the chat function that's here in the webinar presentation, and I'll gather those and ask them after Dr. Stark's presentation. This webinar is being recorded and will be available within the next few days on the AcresUSA YouTube channel. You can find out more about AcresUSA, our magazine, our books, our live events, and more at acresusa.com. Finally, before I turn it over to Dr. Stark, I just want to remind you that there are CEU credits available for this webinar, but you must self-report, and they're only available if you are watching live with us today. So there you go, Dr. Stark.
Okay, thanks, Lydia. Thanks, everyone, for joining. And so we're going to talk about microbes and fertilizer efficiency. So, you know, I give similar talks like this to a lot of audiences. I just want to start off. So I was at the national meeting of AcresUSA, you know, just right there across the river from Cincinnati in December. And generally speaking, the people I spoke with there are about 5 steps in front of most of the people that I work with. So congratulations. So a lot of this you may find doesn't matter as much to you, but hopefully you learn something, because we'll talk about microbes and fertilizer, things I think you already know, and how we can take advantage of these on your farm and your operation. There we go. Now it works too well. So, you know, again, first of all, we have just a big problem with nutrient use efficiency. Maybe I should say nutrient use inefficiency. Now, these are average numbers, so probably not your farm. Depends a lot on your management practices and soil types. But it's definitely fair to say that less than half of the fertilizer we apply goes to the intended crop. You can see for N, P, and K, the relative differences in efficiency. Candidly, 40 to 65% for nitrogen generally is pretty generous. That I see a lot of operations that are less than that. So again, why do we matter? Why does this matter? If we think about, you know, 36% of the operating costs to grow a corn crop is fertilizer in 2021, and the U.S. spends about $20 billion a year in fertilizer. That is, you know, over $10 billion really of your money not going to what you want. So we have to do better. So there are a couple different things that I'm going to talk about. So first of all, I'll talk about N, P, and K and how we get it into the ground. I'll talk about the role of microbes. So just kind of bear with me. So I'll first talk about nitrogen. There are a couple different approaches from a microbial standpoint that people are taking. One is to take advantage of certain bacteria that have the capability of taking nitrogen from the air, and nitrogen is 78% of the air we breathe, and then converting it into ammonia, which can then be used to produce proteins, etc. Now, there's an enzyme called nitrogenase that can do this. And it occurs in bacteria. I can tell you it's extremely energy expensive for bacteria to do this. So not only does it cost a lot of chemical energy to take nitrogen and turn it into ammonia, but the nitrogenase enzyme is poisoned by oxygen. So bacteria have to not only spend the energy, they have to also spend energy getting rid of the air. So I keep talking about the energy. What do I mean? So at the top, you see the two ends joined by three lines. That's nitrogen gas. It's a triple chemical bonded molecule, two nitrogen atoms. It's one of the most stable triple bonded molecules on the planet. So how much energy does it take to break the triple bonds and make single bonds like we see down there with ammonia, single bonds with hydrogen? Well, lightning will do it. So in a thunderstorm, you actually get some free ammonia and nitrate. Lightning is 50,000 degrees Fahrenheit. So that's a little bit of energy. Our process of making synthetic fertilizer, we don't go to 50,000 degrees. Instead, we only go to 750 to 900 degrees Fahrenheit only, with 150 to 250 atmospheres of pressure. That's what it takes to break that nitrogen nut that's triple bonded and make ammonia. And sadly, we all know what happens when the energy that's in ammonia can be used destructively to go back to nitrogen gas. It's explosive. So this is the kind of energy that it takes for bacteria to do it. And the challenge with using nitrogen fixing bacteria is they have to spend a lot of energy excluding nitrogen or excluding oxygen, breaking that nitrogen gas nut that's really tough while they compete with everything else around them. So from a fitness standpoint, they're less competitive. This is a valid approach. Certainly in natural situations, it works. But on the farm, I know there's a lot of products out there. They've got to have a tough technical row to walk down because of the energy requirement. Probably a better way is to protect the nitrogen that's already in the soil, whether that's stuff that we add, and I don't care if it's synthetic, if it's manure, whether animal manure, green manure, cover crops, root exudates, anything. Because what we want is, that is where we've already spent money, and that's where we have huge inefficiency. So if we look at, the center graphic there surrounded in black, when we have soil that is not biologically active, what happens is we put fertilizer down. Again, I don't care what form it's in. The crop uptakes some, the rest either sits in the soil or it's lost. It erodes, it leaches. That's major forms of nitrogen and all nutrient losses, but it's very inefficient. But now there are over 230 published peer-reviewed scientific studies that show if we have biologically active soil, what happens instead is they're on the right, that the biology in the soil, the microbes, eat the fertilizer, eat nitrogen, they grow on it. And I know we're not trying to really fertilize microbes. We ultimately want to fertilize the crop. But since microbes live within half an inch of a root, as long as we have diverse biology, we're cycling that nitrogen out of the microbes back to the crop, to where most of the crop's nitrogen needs actually passes through microbes first. So it's not put ammonia or urea or whatever down on the ground and the plant takes some up and it's lost. It's actually put the fertilizer down, microbes eat it, other microbes eat them, they put it back to the plant. So what we see again is biologically healthy soils are going to produce greater crop yields per unit of fertilizer input. This is where it gets important. And this is where I know, talking with a lot of you, that you get this. When we have too many nutrients in the soil, and again, I don't care about the source, although it's easier to do with synthetic nutrients, that signals the crop, don't put out the exudates, don't attract microbes, you don't need them. This is especially true with excess nitrogen and phosphorus. So what we want is, we don't want an excess of nutrients in the soil, because when we don't have that, the plants actually will farm microbes by secreting root exudates. Microbes mine the soil. So microbes will get nutrients out of the soil the plant can't access and feed them back. We want this cooperation. That's where we get nutrient use efficiency. So that was a nitrogen example. The same for P and K. Again, most soils have a lot of P and K. It's bound. The plant will never access it. You know, just look at the difference between, for example, your P1 and P2 in your soil tests. But microbes can. They can access that total pool. Same deal as we saw with nitrogen. Most of the phosphorus a plant gets fed in a healthy soil passes through the soil biology first. So again, we want the biologically active soils. So, you know, I like to talk about, you know, what to look for in microbial products, because I know there's tons out there, and it seems like every week I'm given one or two new ones from someone that just has questions. So if you're looking at microbial products that can help unlock phosphorus and potassium, that's credible. There's a lot of them out there that can do that. Ones that can take atmospheric nitrogen and fix it, yes, there's a lot of them out there that can do it. Again, they have the handicap of the energy requirement. Another thing you'll see from a lot of microbes are different microbial products that induce rooting. And I've got a lot of guys that tell me, look, I used a product, I saw more roots, I didn't see anything on the yield monitor, so I'm not sure I got a value. You did. Roots are huge to this whole equation, not only building soil, but nutrient use efficiency, because the soil explored within the root mass of any given crop is only about 1%. Most nutrients need to be next to a root to be taken up. So the more roots, more soil explored, better nutrient use efficiency. Also, those roots, if we're doing things right, are putting out exudates. They're attracting, they're farming microbes, which are mining the soil. So we're getting more bang for our buck. When the roots die, they create channels for air and water, just like your crop. All the microbes in the soil that you want in your soil need air and water. Now, there are some that don't need air. Those are the ones you really don't want. And it's roots that contribute much more than anything we leave on top of the ground to building soil structure and organic matter. So roots are hugely important. So again, a quick overview. Biologically active soils with microbes immobilize your nutrients, they keep it in the root zone, and they cycle it back. And we're going to talk about this next. They also are absolutely required, whatever form of fertilizer you're using, especially if it's not a synthetic, if it's a manure, it's cover crops, whatever. It takes a microbe to break that down and release those nutrients to the crop. And also it's microbes and roots that actually produce topsoil, give the soil structure, etc. So I keep talking about microbes, and what I'm going to do, because I like pictures, is I'm going to just show you pictures of the different microbes. These are the different microbe types that are found in soil, from the most abundant, the fastest to grow, what we're probably not deficient in many soils, although we'll have bacteria. So if you do, a Haney test for oxidative burst or a phospholipid fatty acid test, you may see a lot of bacteria. It doesn't tell you if you've got the right kinds of bacteria, but bacteria, fastest to grow, most abundant. A teaspoon of soil can have over 1 billion, thousands of species. And as we go down the list, actinomycetes, fungi, algae, protozoa, nematodes, there's a lot of really beneficial nematodes, ones that do not eat plant roots. They get larger, slower to grow. They're the predators of the things above them. All of these are absolutely required for biologically active and healthy soil. They all play a role. So if we look at bacteria and actinomycetes, so here's pictures. The picture on the top, that's a picture of bacteria. They come in different shapes. They're not always rods. They don't always form chains. Actinomycetes, that's a close-up. They're just a little bit branched, but they're all small. They're very closely related. They do a lot of things. Again, they're critical for soil, like all the other microbes are. They induce rooting. When they consume carbon, they tend to release it as CO2, which is not necessarily bad, but it's not how we really are going to add carbon to the soil. When we till our ground, We break up soil particles, we put more air in the ground. So now bacteria are exposed to food that may have been, surrounded by soil. And again, bacteria being the fastest to grow, get the first and the largest bite of any nutrient in your soil because they're the most abundant, the fastest to grow, most aggressive. They generally live in a state of starvation. So when food is present, they gorge and they multiply. There's a concept here we're going to talk a lot about, carbon to nitrogen ratio, and it's really important. And we can, again, talk about this with other nutrients, but because nitrogen is such a big part of fertilizer, I'm doing the CN ratio. For every nitrogen atom in a bacteria, there's about 5 carbons. As a biologist, I can tell you that is incredibly high. They are nitrogen pigs. They tie it up. By the way, most microbial products people are trying to sell you are just bacteria. How can you tell? Well, if it's a powder, it's probably a narrow slice of bacteria that can form spores, or it can be microencapsulated for a little bit. It could have some fungal spores, although they don't have a long shelf life. If it's a liquid, you can put on the shelf, tightly sealed at room temperature. It's just bacteria. You can't do that with the other microbes because they need air. So those are bacteria, important. Now fungi, though, are, again, now they're different. And I'm not talking about unicellular fungi. Those are yeast. Yeast are great for making beer and wine and bread. But I'm talking about the fungi like in this picture that put out all these long, feathery hyphae that in mycelia. They get in between soil particles that your root hairs can't because they're tiny. They are extremely good at degrading organic matter, especially woody organic matter. So if you have a problem with your corn stalks and your wheat straw, we'll talk later about you need fungi to break it down. Other microbes don't have the biochemistry. All that hyphae and mycelia you see going into the soil, that's carbon rich. So even though they also burn carbon as CO2, they add carbon to the soil. Fungi are really, really good at unlocking phosphorus and finding water. Now their carbon to nitrogen ratio is 3 times lower than bacteria. For every nitrogen, now we're at 15 carbons. So there's something going on here, and they eat bacteria. And by the way, only a few microbial products will contain fungi, and usually only 1 or a couple species. They need oxygen, by the way, too. Now here's some that you probably don't hear much about at all. The protozoa, amoeba, and even beneficial nematodes. So that is a micrograph of a nematode eating another nematode. And there are a lot of species that are beneficial. Now, protozoa, amoeba, nematodes, they're predators. They eat each other, they eat bacteria, they eat fungi, they can eat organic matter. A single protozoa can eat 10,000 bacteria a day. 10,000. Like most fungi, protozoa, amoeba, and nematodes need oxygen. Now their carbon to nitrogen ratio is now 6 times lower than bacteria. So for every nitrogen, 30 carbons. So what's going on here? It's easy. You put fertilizer on the ground. If you have biologically active soil, bacteria eat it first. The bacteria can tie it up because they have a high nitrogen requirement. Their predators, though, as they eat the bacteria, have a lower nitrogen requirement. They have to get rid of it. It's toxic. Just like we get rid of urea when we go to the bathroom. That, what we do kind of sounds like urea, doesn't it? Same deal. So they spit out ammonia and nitrate. What are the 2 forms your plant takes up the most readily? Ammonia and nitrate. Where does all this happen? Within half an inch of a root, because that's where the food lives, the bacteria. The cycle is complete. So again, the importance, the critical nature of biological diversity is so important. And, like I tell people, as a molecular biologist, 20, 25 years ago, I would have said, isn't this just amazing how evolution did this over millions and millions of years? And this has become a passion of mine because I was so wrong. Because as I've really looked into the data, this is design. This is, mathematicians were laughing at me saying, Dave, what you're proposing is absurdly impossible. And I guess that's a real mathematical term. This is design, and I believe it's God's design. And again, that's something else that if you want to talk more about, I'd be happy to. But this is the way soil was designed to be. So people will ask me, if I have biologically active soil, do I have to keep using microbial products? And the answer is generally yes. So we just heard about all the snow and how cold it is in Colorado. That's killing a lot of the soil microbes. Microbes are full of water. What happens to water when it freezes? It expands. Microbes have cell walls, which are fairly rigid, so they explode. Now, we don't sterilize the soil when we freeze it, but we do knock it back, and we don't do it evenly. So some microbes are better freeze tolerant than others, so the soil is now out of whack. Flooding is another thing we have no control over. After about 48 hours underwater, the oxygen underneath is depleting, so now we're enriching for the bacteria that can survive without air. Those tend to be the plant pathogens, and the beneficials that need air are getting knocked way back. So the soil is out of whack. Again, I've already said tilling, overuse of chemicals, fertilizer, fumigants, all knocks back soil microbes. But just to make this even more complicated, we're finding now that it's not just differences between corn, soy, wheat, a cherry tree, an oak tree, a flower, that microbes are specific to different kinds of plants. 6 to 10% of the soil biome varies between corn hybrids, between soybean varieties. So the layer of complexity just got even higher in terms of how picky plants are in farming the microbes they want, excluding the ones they don't. So again, we need that diverse population so that we meet the best optimal needs of what that plant is. So, I mean, we have a particular product. I'm not a sales guy. I don't like doing sales pitches. I'm a scientist. But you want products like this that have diverse biology. So ours is based off of, the old way of making compost tea. So compost teas can be a very good product that gives diverse biology. I really like compost teas. But if you find that they're a pain, they take too much time, they're inconsistent batch to batch, what we make is an engineered compost tea. We have a very precise starting inoculum. We change conditions throughout the brew. We DNA fingerprint at the end. So we know we have over 600 species of bacteria, hundreds of species of fungi and protozoa, amoeba, beneficial nematodes. We put in food sources like molasses, amino acids, a little humic and fulvic acid, kelp extract, and we stabilize all this through refrigeration. So if someone's selling you what they claim is a diverse biological product and it's not refrigerated, it's not diverse. Because I'm telling you, if I could invent a way to get away from refrigeration, we'd do it. We've tried, we're still trying. It's the best way though to keep living biology, not spores, because most of what you need in the soil doesn't form spores. Living biology is stable. So, let's look at some of the evidence, and I'll go a little bit quickly here, just some of the data. So this is, using our product. You could get something, again, similar with a really good aerobic compost tea. But this is a summary of 26 different studies, North Carolina State, Penn State, Purdue, on turf. Turf as a grass, like corn and wheat and barley, et cetera. And what we're looking at is turf performance with different levels of nitrogen input, different forms of nitrogen, very different soil types and climates. And you can see massive reductions possible. In no way does any biology influence the plant so it needs less nitrogen. In every way, what's happening is the nitrogen that's put down stays in the root zone and gets cycled back to the plant. So more bang for the buck. And that's what we're seeing. By the way, you're not going to reduce your nitrogen 75 or 88% in the crop because we remove nutrients when we harvest. My whole point is it's a tool to help you dial in your inputs that maximizes efficiency in your operation. So now this is corn. This is in Illinois, so really good ground. The first test, this is in the bar graph. This is a third-party researcher, by the way, coming off of soybeans, a lot of nitrogen in the bank. He only had to put down 45 pounds of UAN to get his target 230 bushels an acre. So again, really rich ground. Here we wanted to do 20% less. That's only 11 pounds. No impact on the yield with our biology. It did have an impact without our biology, but when we kept it at 45 pounds, he got 11 bushels more. Again, 45 pounds is low. We're not bombarding the root zone with nutrients. The plant is still hungry enough to feed microbes. Now, the following year, we're corn on corn. Now it's 180 pounds of nitrogen. A 20% reduction is a lot more significant there. 20% less one, it got 9 bushels more. My goal, my point, isn't to give a formula for how to use a product like this. It's again to get you to think about using diverse soil microbes as a tool that helps you with what you're doing, get the best out of your efficiency. We did other trials this last summer in Iowa. And again, you can see 5 to 20% reduction. By the way, all the fertilizer reductions that I talk about happen either before planting or at the planter. If the crop during the season is telling you it needs certain nutrients, I think you should feed the crop those nutrients. This is more the plant and pre-plant. And here you can see 20% less fertilizer. We still got about a bushel more. Probably pencils out better though to only back off 5% because of the price of corn and such. But again, it's a tool. This is a pasture in Alabama, reseeded with a fescue mix and watered. This is the same field. On one part of the field, only water, the other part of the field, our biology. And look at the difference. So what's it showing? It's showing that, first of all, this pasture doesn't have much biological activity because without adding biology, there are not many nutrients being released. But we put diverse biology, now the grass is being fed. It's green and it's growing. It works. The biology actually does all this. This is West Central Indiana, right on the Illinois border. Potassium is a major nutrient that gives strength to stalks and stems. And a lot of the microbes, again, keep potassium from getting tied up. They unlock it, they feed it to the plant. And you can see here just the difference between the two stalks and the strength. And we hear this all the time. The stems are stronger, the stalks are stronger, the brace roots are bigger and stronger. More nutrients will reward you with a healthier plant and usually higher yield, as we see here. Now, I love this picture, and I'm going to spend a minute, and I know I'm getting really close to being over, but I like it because it's a free test. So here we're comparing in the middle. This is a nitrogen-fixing bacteria. It's free living, so it's not in the plant, so it's not like rhizobia. This is free living from a company you've probably all heard of. On the right is just starter fertilizer. The left three are our product, the nitrogen-fixing bacteria, and the starter fertilizer. So this picture tells me a lot. Now, what the grower noticed is the plants with all three were deeper green, they were taller, the roots were more extensive and a lot more root hairs. And this is, again, a free test. Look at the middle picture. There's no dirt on the roots. There aren't many root hairs. The same with the right, with just the starter fertilizer. So what's going on? You see, this is telling me this ground is not biologically active, because for the dirt to stick to the roots, we need quorum sensing. We need not only a level of microbes in the soil, a diversity of microbes, because each species has metabolites or biochemical signals they send out. They sense the signals from each other. When they sense that there's a critical mass of numbers and types, they change their gene expression. They start working with the root exudates and they make a glue, and they glue the dirt to the roots. This is where all the action happens. Nutrient exchange, nitrogen fixation, water retention. It also triggers the plant to start taking up microbes at the root tip. called rhizophagy. They actually eat microbes in the root tips. They strip off their cell walls for the nitrogen. Some of the microbes die, so they get all the nutrients. Others, though, travel up the root till they get the bud for the root hair. They secrete a hormone that induces the root hair formation. They travel down the root hair. They're spit back out in the soil. They remine the soil for nutrients and repeat the process. So you can see biological activity. We're getting more root hairs, we're getting more roots, and we're getting dirt glued to the roots. It's just this test, all you needed was a shovel, and you could see the difference in what a biologically diverse product will do. So last, I'll go quickly. You probably do a lot of keeping crop residue on the field. That's fantastic. The carbon is important, but there's also a lot of nutrients you've already paid for. So in corn, every ton of corn stover has 17 pounds of nitrogen, 4 pounds of phosphorus, 34 potassium. Remember, it's an important part of the strength of the crop. And a 200 bushel an acre crop has 5 tons. So we want to get those out. When you're looking at a product to break down residue, we're back to carbon and nitrogen ratio. Corn stover has 1 nitrogen for about every 60 carbons. Wheat is 1 to 80. So don't use a product that's just bacteria that has a 1 to 5 requirement, because the yellow corn can happen. It'll tie up your nitrogen. You need fungi. So this is a product, again, one of ours, that is rich in the fungi that can break down that crop residue. This is 3 weeks. Ideal conditions, though, because they had temperature and moisture. Only microbes break down crop residue. Microbes like your crop need temperature and moisture. So, would you go out today in Colorado where it's 0 degrees and all the snow? No. You've got to do this when you still are going to have a few weeks of really 40 degrees or more, or sunny days that can warm up the soil with some moisture, even morning dew under the mat to break this stuff down. Otherwise, you probably just wait till spring. So I threw a lot at you. Just remember, if you really want products that are going to help you boost your soil and your nutrient use efficiency, go with biological diversity. Bacteria, fungi, actinomycetes, protozoa, amoeba, and feed them, do everything else you're doing, because a microbial product, including ours, nothing is a standalone intervention. It all works with other things you're doing, reducing your tillage, putting more carbon in the ground, all the other things that you do to take care of your ground. This is just a component. So with that, Lydia, I will stop and take questions. I think I'm just a couple minutes over.
Thank you so much, Dr. Stark. That was perfect timing, and you covered so many important points so well. We've gotten some really great questions here, and I think we're going to start with a big one, which is very specific, though. It says, once established and without damage from freezing or tillage, how long will the biological activity, such as mycorrhizal fungi, live in the soil?
So mycorrhizae that's really a unique case, because mycorrhizae need a living root. And when they don't have one, now they will form spores and they can overwinter. But what I haven't seen is really good data that can answer your question specifically. So if I don't till my ground, and tilling is one of the worst things you do specifically for mycorrhizae, will they overwinter? Do I need to add them every spring? And I suspect it just depends. If you're in a place where you're not getting flooding, you're not getting a really hard, deep freeze, probably not. But again, I'm a scientist. I want to see data. And this is where I'm telling you, this is my impression versus based on hard data. I do know there are studies that look at biology, and the microbiome in the soil literally changes week to week through your growing season. And we don't understand all the changes and why. Someday, I predict, seed companies that will figure this out, they will sell you genetics, and they will sell you a microbe kit that is ideal for those genetics. We're not there today. So And I'm not afraid to tell you, the more I know, the dumber I think I get. So, because there's so much out there we still don't know in the soil.
Thank you so much, Dr. Stark. Here's a question I suspect you do know the answer. Farouk asks, what form is the nitrogen in the microbes that then goes to the plants?
So they're going to use it, they'll mostly protein. And when other microbes eat them, they're getting a high dose of protein. So it's like us, eating, a really large meat meal. We've got a lot of nitrogen to get rid of. So their predators, the fungi, protozoa, and amoeba, take the protein, they're breaking it down into ammonia and nitrate, and that's what they expel.
Got it. He has a second follow-up question about trace elements like zinc or copper and asks if they go through the same route.
Yeah, they do, because again, there are microbes in the soil that secrete chelators of all of those, the metals and organic acids and such, so they can unlock all of them. And the biology in the soil has really similar nutrient requirements as your crops. So they also need iron and zinc and magnesium and manganese, et cetera, and calcium. So when they are dying or they're going into the root through rhizophagy, they're giving those nutrients up and feeding the plant.
Great, great. We also have a question that's a little bit bigger, which asks about synthetic nitrogen and the use of synthetic nitrogen and why it's frowned upon in the organic market. Jennifer says she can't get an organic certificate because she uses synthetic nitrogen, although she doesn't use any other harmful chemicals.
Well, I think the idea of being organic and using inputs that only come from nature, is why a synthetic nitrogen fertilizer is not allowed. Now, if you go to the USDA site, there are a lot of exceptions in terms of synthetic substances that are allowed. I mean, ideally, if what we really want to do is take care of our soil, personally, I don't think there's anything wrong with synthetic nitrogen. I think actually it's been a blessing. It just makes it easy to over apply. And that's where we have to start backing off, because we're not helping ourselves and our soil. But that's why, because it's something synthetic. So the people that came up with the organic USDA regulations excluded it.
So it's a function of the regulatory process at the time, and maybe
Yes.
now as there's revisiting some of these standards, maybe some of these aspects of it will also be revisited.
And I hope so, because I've done a lot of studies on organic agriculture, and nitrogen is one of the most limiting nutrients because it's so hard and so expensive if you can't go synthetic. So if anything, an organic farmer has to have biologically active soils to get the most out of what they can access.
Sticking up with nutrient application, not nitrogen, turning to nutrient, I should say. David asks, with regard to nutrient efficiency, is in-row application more or less efficient than broadcasting?
Well, I hear a lot of people talk both ways. So when it comes to nitrogen, we know if we go on the seed that nitrogen can burn. And so some crop types are more tolerant to that than others. I think corn is a little more tolerant than beans. Plus, you don't want to put nitrogen on a soybean that signals the bean don't soak up the rhizobia to do nitrogen fixation. So again, we want to keep those separate. If I look again at nitrogen losses, erosion and runoff are two of the big causes, much more than nitrification, for example. So if you put it on the ground, what I really don't like is fall nitrogen application. To me, that's a lot of your money, and a lot of things can happen over the winter to where that nitrogen is just not going to be there. Otherwise, I think it makes more sense to put it where the roots are going to grow. So I like 2x2. I like the wide drops, but I haven't seen enough data that says if you're doing one versus the other, you're really making a big mistake.
Great. That's really helpful answer. Turning to the Holganix product itself, Roger asks, how long is your product viable after it is taken out of refrigeration?
Depends on the container size and the outside temperature. So most farmers are buying in a 250-gallon tote. They get 4 to 8 weeks. So it's not ice cream. The bigger problem is if you mix it in a tank with fertilizer, especially nitrogen, and you hit a long weather delay, they're living microbes. They're going to grow in the tank, and they're going to clog your filters coming out. They won't after a day or two, but if you're in a week or more, that's where there's nothing we can do. But otherwise, it's very rare that the product goes bad. And when it goes bad, it goes from smelling kind of sweet and earthy to swamp water where something died and is half decayed. I mean, it's just disgusting. When that happens, we always replace it at our expense. And I think I've done that twice in the last 3 years. So we have pretty good stability.
What about appropriate mixes? It seems like it might be possible to identify which microbes are preferred by specific crops. So are you likely to consider appropriate mixes for the major crops?
You know, we've looked at that, and it's something that we'll continue to do. So for example, we can take microbes from, corn hybrid A, through the root zone. We take them all because there are microbes that live in and on the plant, even above ground. What I haven't seen is enough difference in performance that it would warrant the extra expense of making specific products. And I think what that means is we haven't learned enough how to make that product really, really different and better. So right now, just going with something broadly diverse like we have seems to be what it's not expensive and generally works well everywhere we use it. But we'll get smarter as time goes on. I'm just not there yet.
Maybe coming to the end, we could ask a bigger question that came up right at the beginning, which maybe takes a tough, complicated answer, but maybe you could simplify it, was how do you measure soil health?
That is a fantastic question. And if you're part of the Soil Health Institute like I am, we've had those debates. And I've seen different definitions that really are getting there. But to me, I look for a few things. pH. pH is really important. If you're, 6 or below or 8 and above, then you're tying up nutrients that's not ideal for your roots or the biology. Generally speaking, I mean, there are some acid-loving plants, et cetera. We want organic matter, but the more isn't always the better. So a 20% organic matter, now you're like a bog and you're going to hold too much water. 0.5% is like the beach, or come on, it doesn't hold anything. So we want organic matter and we want carbon in there, and we want cation exchange capacity, and that's the ability of the soil to hold on to nutrients, especially. I'm going to nerd out divalent cations. So what are those? Iron, magnesium, calcium. The important nutrients, ammonia. So anything with a positive charge, and keep it from getting tied up in the soil. So we have a good mix of all of those things with good biological activity. Because again, it's the biology that makes soil work.
Fantastic, fantastic. Well, listen, this has been an amazing masterclass. I really want to thank you, Dr. Stark, on behalf of AcresUSA, and remind all our participants that this recording will be available for you to review, and we will take all your questions and present them to Dr. Stark later on for response. So Hannah, I'm going to hand it back to you, I think, to close us out.
Yeah, all right. Dr. Stark, thank you so much for coming. As always, this was incredibly informative. And is it okay if people email you their questions? I saw you had your email on your slide there.
Yes,
yeah,
absolutely.
Perfect. So yeah, if you were unable to get your questions answered, please feel free to shoot Dr. Stark an email and he'd be happy to answer it for you there. I know we had a ton of questions. We weren't able to get to all of them. So thank you guys for being a super receptive audience and for making this another great webinar.
Thanks, everyone. Thank you.
Bye-bye.
Yeah, bye-bye.
All right. Bye, everyone. Thank you all so much for coming. Thank you for your incredible questions. And as I said, please feel free to shoot Dr. Stark an email if you were unable to get your question answered before the end. All right.